Chapter 1, Section 2: How Scientists Work. Have you ever noticed what happens to food that is left in an open trash can for a few days in the summer? Creatures that look like worms appear on the discarded food. These creatures are called maggots. For thousands of years, people have been observing maggots on food that is not protected. The maggots seem to suddenly appear out of nowhere. Where do they come from? About 2,300 years ago, the Greek philosopher Aristotle made extensive observations of the natural world. He tried to explain his observations through reasoning. During and after his lifetime, people thought that living things followed a set of natural rules that were different from those for non-living things. They also thought that special vital forces brought some living things into being from non-living material. These ideas, exemplified by the directions in Figure 1-7, persisted for many centuries. About 400 years ago, some people began to challenge these established ideas. They also began to use experiments to answer their questions about life. Now look at Figure 1-7. About 200 years ago, a Roman poet wrote these directions for producing bees. Recipe for bees. Number 1. Kill a bull during the first thaw of winter. Number 2. Build a shed. Number 3. Place the dead bull on branches and herbs inside the shed. 4. Wait for summer. The decaying body of the bull will produce bees. Asking a question. For many years, observations seem to indicate that some living things could just suddenly appear. Maggots showed up on meat, mice were found on grain, and beetles turned up on cow dung. People wondered how these events happened. They were, in their own everyday way, identifying a problem to be solved by asking a question. How do new living things or organisms come into being? Forming a hypothesis. For centuries, people accepted the prevailing explanation for the sudden appearance of some organisms, that some life somehow arose from non-living matter. The maggots arose from the meat, the mice from the grain, and the beetles from the dung. Scholars of the day even gave a name to the idea that life could arise from non-living things: spontaneous generation. In today's terms, the idea of spontaneous generation can be considered a hypothesis. In 1668, Francesco Redi, an Italian physician, proposed a different hypothesis for the appearance of maggots. Redi had observed that these organisms appeared on meat a few days after flies were present. He considered it likely that the flies laid eggs too small for people to see. Thus, Redi was proposing a new hypothesis: flies produce maggots. Redi's next step was to test his hypothesis. Setting up a controlled experiment. In science, testing a hypothesis often involves designing an experiment. The factors in an experiment that can change are called variables. Examples of variables include equipment used, type of material, amount of material, temperature, light, and time. Suppose you want to know whether an increase in water, light, or fertilizer can speed up plant growth. If you change all three variables at once, you will not be able to tell which variable is responsible for the observed results. Whenever possible, a hypothesis should be tested by an experiment in which only one variable is changed at a time. All other variables should be kept unchanged or controlled. This type of experiment is called a controlled experiment. The variable that is deliberately changed is called the manipulated variable. The variable that is observed and that changes in response to the manipulated variable is called the responding variable. Based on this hypothesis, Reddy made a prediction that keeping flies away from the meat would prevent the appearance of maggots. To test this hypothesis, he planned an experiment shown in figure 1-8. Notice that Reddy's controlled all the variables except one, whether or not there was gauze over each jar. The gauze was important because it kept flies off the meat. Now look at figure 1-8. In a controlled experiment, only one variable is tested at a time. Reddy designed an experiment to determine what caused the sudden appearance of maggots. In his experiment, the manipulated variable was the presence or absence of the gauze covering. The results of this experiment helped disprove the hypothesis of spontaneous generation. In your notes, go ahead and sketch out Reddy's experiment on spontaneous generation. Observations: flies landed on meat that is left uncovered. Later, maggots appeared on the meat. His hypothesis: flies produce maggots. He had uncovered jars, maggots appeared. He had covered jars, no maggots appeared. The controlled variables were jars, type of meat, location, temperature, time. The manipulated variable, gauze covering that keeps flies away from meat. The responding variable, whether maggots appeared. The conclusion was maggots form only when flies come in contact with meat. Spontaneous generation of maggots did not occur. Recording and analyzing results. Scientists usually keep written records of their observations or data. In the past, data were usually recorded by hand, often in a notebook or personal journals. Sometimes, drawings such as a figure in 1-9 recorded certain kinds of observations more completely and accurately than a verbal description could. Today, researchers may record their work on computers. Online storage often makes it easier for researchers to review the data at any time and, if necessary, offer a new explanation for the data. Scientists know that Reddy recorded his data because copies of his work were available to later generations of scientists. His investigation showed that maggots appeared on the meat in the control jars. No maggots appeared in the jars covered with the gauze. Drawing a conclusion. Scientists use the data from an experiment to evaluate the hypothesis and draw a valid conclusion. That is, they use the evidence to determine whether the hypothesis was supported or refuted. Reddy's results supported his hypothesis. He therefore concluded that the maggots were indeed produced by flies. As scientists look for explanations for specific observations, they assume that the patterns in nature are consistent. Thus, Reddy's results could be viewed not only as an explanation about maggots and flies, but also as a refutation of the hypothesis of spontaneous generation. Now, look at figure 1-9. For centuries, the workings of the human body remained a mystery. Gradually, scientists observed the body's structures and recorded their work in drawings like this. This diagram dates back to the 15th century in Austria. Repeating investigations. A key assumption in science is that experimental results can be reproduced because nature behaves in a consistent manner. When one particular variable is manipulated in a given set of variables, the result should always be the same. In keeping with this assumption, scientists expect to test one another's investigations. Thus, communicating a description of an experiment is an essential part of science. Today's researchers often publish a report of their work in a scientific journal. Other scientists review the experimental procedures to make sure that the design was without flaws. They often repeat experiments to be sure that the results match those already obtained. In Reddy's day, scientific journals were not common, but he communicated his conclusions in a book that included a description of his investigation and its results. Needham's test of Reddy's findings. Some later tests of Reddy's work were influenced by an unexpected discovery. About the time Reddy was carrying out his experiment, Anton van Leeuwenhoek of the Netherlands discovered a world of tiny moving objects in rainwater, pond water, and dust. Inferring that these objects were alive, he called them animalcules, or tiny animals. He made drawings of his observations and shared them with other scientists. For the next 200 years or so, scientists could not agree on whether the animalcules were alive or how they came to exist. In the mid-1700s, John Needham, an English scientist, used an experiment involving animalcules to attack Reddy's work. Needham claimed that spontaneous generation could occur under the right conditions. To prove his claim, he sealed a bottle of gravy and heated it. He claimed that the heat had killed any living things that might be in the gravy. After several days, he examined the contents of the bottle and found it swarming with activity. These little animals, he inferred, can only have come from juice of the gravy. Spallanzani's test of Reddy's findings. An Italian scholar, Lazzaro Spallanzani, read about Reddy's and Needham's work. Spallanzani thought that Needham had not heated his samples enough and decided to improve upon Needham's experiment. Figure 1-10 shows that Spallanzani boiled 2 containers of gravy. Assuming that the boiling would kill any tiny living things or microorganisms that were present, he sealed one jar immediately and left the other jar open. After a few days, the gravy in the open jar was teeming with microorganisms. The sealed jar remained free of microorganisms. Spallanzani concluded that non-living gravy did not produce living things. The microorganisms in the unsealed jar were offspring of microorganisms that had entered the jar through the air. This experiment and Reddy's work supported the hypothesis that new organisms are produced only by existing organisms. Now take a look at figure 1-10. Spallanzani's experiment showed that microorganisms will not grow in boiled gravy that has been sealed, but will grow in boiled gravy that is left open to the air. What variable was controlled in this experiment? In your notes, go ahead and sketch Spallanzani's experiment using figure 1-10. Pasteur's test of spontaneous generation. Well into the 1800s, some scientists continued to support the spontaneous generation hypothesis. Some of them argued that air was a necessary factor in the process of generating life because air contained the life force needed to produce new life. They pointed out that Spallanzani's experiment was not a fair test because air had been excluded from the sealed jar. In 1864, an ingenious French scientist, Louis Pasteur, found a way to settle the argument. He designed a flask that had a long curved neck, as shown in figure 1-11. The flask remained open to the air, but microorganisms from the air did not make their way through the neck into the flask. Pasteur showed that as long as the broth was protected from microorganisms, it remained free of living things. About a year after the experiment began, Pasteur broke the neck of the flask, and the broth quickly became filled with microorganisms. His work convinced other scientists that the hypothesis of spontaneous generation was not correct. In other words, Pasteur showed that all living things come from other living things. This change in thinking represented a major shift in the way scientists viewed living things. Take a look at figure 1-11. Pasteur's experiments showed that boiled broth would remain free of microorganisms even if air was allowed in, as long as dust and other particles were kept out. In your notes, go ahead and sketch out Pasteur's experiment. The impact of Pasteur's work. During his lifetime, Pasteur made many discoveries related to microorganisms. His research had an impact on society as well as on scientific thought. He saved the French wine industry, which was troubled by unexplained souring of wine, and the silk industry, which was in danger by a silkworm disease. Moreover, he began to uncover the very nature of infectious diseases, showing that they were the result of microorganisms entering the bodies of the victims. Pasteur is considered one of biology's most remarkable problem solvers. Biology and history. Major discoveries. The history of biology includes discoveries about the structure of the human body, the nature of cells, how species evolve, ways to fight deadly diseases, and what molecules determine hereditary traits. You will learn about these discoveries as you study this textbook. 1543, Andreas Vesalius. Vesalius published On the Structure of the Human Body, the first accurate and detailed study of human anatomy. 1628, William Harvey. Harvey is the first scholar to describe the circulation of blood. He shows how blood pumped through blood vessels returns to the heart and is recirculated. 1673, Anton van Leeuwenhoek. Van Leeuwenhoek perfects the simple microscope and observes cells and microorganisms. 1859, Charles Darwin. Darwin publishes On the Origin of Species, stating that all forms of life have evolved into their present state over the course of millions of years. 1881, Louis Pasteur. Pasteur develops the first vaccine against anthrax, a deadly bacterial disease. 1953, James Watson and Francis Crick. Watson and Crick determine the structure of DNA. When experiments are not possible. It is not always possible to do an experiment to test a hypothesis. For example, to learn how animals in the wild interact with others in their group, researchers carry out field studies. It is necessary to observe the animals without disturbing them, as shown in Figure 1-12. Ethical considerations prevent certain experiments, such as determining the effect on people of a chemical suspected to be causing cancer. In such cases, medical researchers may choose volunteers who have already been exposed to the chemical. For comparisons, they would study a group of people who have not been exposed to the chemical. When researchers design such alternative investigations, they try to maintain the rigorous thinking associated with a controlled experiment. They often study large groups of subjects so that the small differences do not produce misleading results. They try to identify as many relevant variables as possible so that most variables are controlled. For example, in a study of a cancer-causing chemical, they might exclude volunteers who have other serious health problems. By exerting great care in planning these kinds of investigations, scientists can discover reliable patterns that add to scientific knowledge. How a theory develops. As evidence from numerous investigations builds up, a particular hypothesis may become so well supported that scientists consider it a theory. That is what happened with the hypothesis that new organisms come from existing organisms. This idea is now considered one of the major ideas in science. It is called biogenesis, meaning generating from life. You may have heard the word theory used in everyday conversations as people discuss ideas. Someone might say, "Oh, that's just a theory," to criticize an idea that is not supported by evidence. In science, the word theory applies to a well-tested explanation that unifies a broad range of observations. A theory enables scientists to make accurate predictions about new situations. Sometimes, more than one theory is needed to explain a particular circumstance. For example, why are the marsupial mammals in Figure 1-13 found only in Australia and some nearby islands? An answer lies with the theories of plate tectonics and evolution. Millions of years ago, when marsupials were evolving, Australia, Antarctica, and South America were joined as a single landmass. That landmass began to break apart, and Australia became a separate continent. Its marsupials were thus separated from other kinds of mammals, and they evolved as a unique group. You will study the theory of evolution in Unit 5. A useful theory may become the dominant view among the majority of scientists, but no theory is considered absolute truth. Scientists analyze, review, and critique the strengths and weaknesses of theories. As new evidence is uncovered, a theory may be revised or replaced by a more useful explanation. Sometimes scientists resist a new way of looking at nature, but over time, new evidence determines which ideas survive and which are replaced. Thus, science is characterized by both continuity and change.